We continue now talking about the processing of eukaryotic pre-mRNAs by discussing the modification that occurs in the region between the 5' cap and the 3' poly A site. Therefore, I term this the middle modification to a pre-mRNA. That modification is mRNA splicing. Comparing a bacterial gene to a eukaryotic gene, we see the eukaryotic gene on the bottom contains coding regions that will ultimately encode for a protein and be in the mature mRNA, and non-coding regions, identified here as introns, regions that will be removed from a pre-mRNA and will not be part of the mature mRNA. The eukaryotic DNA then is transcribed to produce a pre-mRNA that contains both the coding regions and the non-coding regions. However, the non-coding intronic regions will be excised or removed, and the coding regions, the exons, will be spliced together to create the mature mRNA that will ultimately be translated to create the protein. All eukaryotic genes that I'm aware of contain both exons and introns. However, the number or the relative number of introns and exons is not always the same. Here we see two human genes. The beta-globa gene has 3 exons that ultimately will be spliced together, and the gene itself is quite small. Compare that to the human factor VIII gene that is quite large, 200,000 nucleotides within that gene, and within those 200,000 nucleotides, 26 different exons that ultimately will be spliced together to create the mature mRNA. While it may seem inefficient to transcribe a lot of mRNA that ultimately will not remain within a mature mRNA, you learned the rationale for this system in the summary video. However, I'll reiterate it here with this hypothetical gene that contains 3 different exons. Of course, each time this hypothetical gene is transcribed, a pre-mRNA, shown in the middle, will be produced, and that pre-mRNA will always contain all 3 exons. However, dependent upon how the introns are removed, it's possible that this hypothetical gene might produce 3 different mRNAs, and each of those mRNAs would ultimately produce a different protein. In this way, by alternatively splicing the pre-mRNA that is produced from a gene, we can have 3 different proteins produced from a single gene. Therefore, it's conservation of space within our genomes. The process of pre-mRNA splicing is a little complex, so the next couple of slides may get a little messy, and you may have to view them a few times to be clear on the process. Here's a summary showing you that it really is a 2-step process to remove an intron. The first step is a transesterification reaction, and that just means that you are moving the ester bond between a couple of different nucleotides in the pre-mRNA. That first step depends upon an A nucleotide within the intron that we refer to as the branch point A. The branch point A is critical in the process of splicing. The second step is also a transesterification reaction where the hydroxyl group that is associated with the 3' end of the 5' exon that hydroxyl group will react with the phosphate group that is on the 5' end of the 3' exon. I'll show you that again. However, I want to point out the sequences that are going to be required for intron removal. I'll also point out that in all of the images that I'm showing you in this process, we're going to keep the same color coding. The exons are always going to be identified in blue, and the introns are always going to be identified in yellow. The branch point A is always going to be highlighted as red. A couple of important sequences that are necessary for intron removal are at the exon-intron borders. The border between the 5' exon and the intron that will be removed is always an AG-GU, and the break will come between the two G nucleotides. The second sequence that's important is at the border between the intron and the downstream exon. That sequence is always going to be an AGG. And again, the break will occur between the two G nucleotides. Of course, we're also going to highlight that branch point A. And I'll show you that at the bottom, when the intron is removed, we will always splice together an AG from the upstream exon to the G from the downstream exon. Okay, here's where it gets a little ugly. On the left-hand side, you have the cartoon that we've seen before, and on the right-hand side, we've got the the mRNA sequence outlined with the chemistry shown. Again, keeping that color coding, the nucleotides that you are seeing are all part of the intron with the branch point A highlighted in red. I will give you some additional reference points as I describe the chemistry that's going to be occurring. In step 1, that first transesterification reaction, the branch point A, shown in red, will interact with the 5' end of that G nucleotide of the intron, and it will do that using its 2' hydroxyl group. In other words, the branch point A is critical because it will be bonded with two different nucleotides, one through its 3' OH and another through its 2' OH. Note that the 2' OH, or the 2' O bond, is going to be with the 5' end of that intron sequence. Again, that nucleotide is identified as a G. To orient yourself within this figure, there's an asterisk that I've highlighted that is upstream of the branch point A, and I've put a little T in the nucleotide that is downstream of that branch point A. The second transesterification reaction is going to be between the hydroxyl group that is on the 3' end of the upstream exon, and that hydroxyl group will interact with the phosphate group that is on the 5' end of the downstream exon. Therefore, the bond between the G in the intron and the G in the exon will be broken as a new bond is formed between the G in the upstream exon and the G in the downstream exon. These are the two trans-esterification reactions associated with mRNA splicing. Proper splicing requires that small nuclear RNAs or snRNAs recognize those important sequences, at the intron-exon border and the branch point A. There are two critical snRNAs, one called U1 snRNA. U1 snRNA recognizes the sequence that AG, GU, and surrounding sequences associated with that upstream exon / intron border. A second small nuclear RNA, U2, recognizes the branch point A. These small nuclear RNAs are associated with proteins. The combination of an snRNA and its associated proteins creates what we call an snRNP, or a small nuclear ribonuclear particle. We have U1 snRNP and U2 snRNP, and that's a mouthful. So rather than saying U1 snRNP, we say U1 SNRP and U2 SNRP. Here's a cartoon showing you how the SNRPs facilitate splicing. We have our U1 and our U2 SNRPs with the blue snRNAs, the heavy blue lines depicting the snRNAs, and the colored green and blue respective blobs surrounding those snRNAs to represent the SNRP. U1 and U2 will bind to the important sequences that they're supposed to recognize, and another triple SNRP, U4, 5, 6 triple SNRP, is responsible for bringing the U1 and the U2 SNRPs together in space such that the trans-esterification reactions required for splicing are able to occur. Once splicing has occurred, U1, U2, and the excised intron in the form of what we call a lariat, so this is a lariat like you are going to be lassoing cattle, with the branch point A being sort of that knot in the lariat. That lariat is removed with U2 SNRP, and ultimately that intron is degraded and the ribonucleotides are recycled into a new pre-mRNA. All of this splicing is occurring while transcription is continuing, and in fact, after transcription has occurred. There is a lot of activity going on along with transcription, and here you see a hypothetical pre-mRNA with associated U1 and U2 SNRPs, and a whole bunch of other things that orchestrate this whole splicing process. To summarize, there are three modifications that are associated with the processing of a pre-mRNA. Those include the addition of a 5' methyl G cap, the addition of several hundred adenosine nucleotides in the form of a poly A tail at the 3' end, and removal of intronic sequences through the process of mRNA splicing. Once processing of a pre-mRNA is complete, proteins associate with the cap and the poly A tail, and those proteins are involved in the movement of the now mature mRNA from the nucleus to the cytoplasm. Note that the protein that is associated with the 5'-methyl G cap, once in the cytoplasm, is removed and it is replaced by a different cap binding protein known as an initiation factor that will facilitate or make more efficient the process of translation.